Welcome everybody. So it's wonderful to see an almost full lecture theatre. I'd like to acknowledge the traditional owners of the land on which we're meeting here today, the Wurundjeri people of the Kulin Nation, and pay my respects to their elders past and present. I'd also like to acknowledge all of the Jansz family that's here in the front row and many friends. It's really great to see lots of people here to support Tash. So Tash I've known for a number of years because she started by doing honours with James Murphy and I in 2013. And today she's going to talk about 2 of her 3 PhD projects that have been incredibly successful and that will end up resulting in 2 really great first author papers. And I think what's really most striking about these projects is that they've shaped our vision about how Smchd1, one of the epigenetic regulators that we work on, how it actually works at the chromatin, what it's doing to switch genes off. And this is a big change for what a PhD student can do within the lab. So the reason that she's been so successful and that she's such a brilliant mind is that she's passionate about what she does, and you'll see that when she's presenting today. She's committed, and so she works really hard at addressing the questions. She's insightful, she reads the literature broadly. She does everything you'd expect of not just a PhD student, but a scientist well beyond her years. So really, I think, though, the thing that has made her such a brilliant PhD student is that she's scientifically fearless. So rather than saying, oh, I'm not sure that's a new technique, Tash will always bite off more than I would think she can chew, or I would think anybody can chew. She deliberately challenges herself to do the hardest thing possible and therefore ask the most challenging questions, and that's why she can really change how we view this particular epigenetic modifier. So, for example, she chose to take on projects that she'll describe today using HoxB to look at chromosome conformation. And in the equivalent of nose-to-tail eating, Tash does nose-to-tail science, where she not only thinks up the experiment, she then does the experiment and takes it all the way through to doing all of the bioinformatic analysis as well. So this is remarkable for somebody at her stage of career, and remarkable that she's not only doing simple analyses out the other end, she's doing analysis of HoxB data sets, which are challenging, I think, for everybody, even really talented bioinformaticians. So beyond this really astounding scientific understanding that she's developed over the last few years, the other thing that I wanted to comment on is that she really makes the lab a better place. So she's simply a pleasure to have around. I don't think there's ever been a single conflict between the 2 of us, which has been delightful, I have to say, as a PhD supervisor. There's been nothing to worry about along the way. She's a generous collaborator, she's empathetic and kind, and she's really a wonderful person to have in the lab environment. So well done, Angela and Jeff, for making such a wonderful, wonderful daughter, and we're going to be very sad to see her go when she does end up going to Munich for a postdoc. But I'm sure that after seeing the seminar today, in a few years' time, we will be welcoming her back again to tell us all about that wonderful postdoctoral study as well. So welcome, Tash.
Right, well, thank you so much, Moni. That was so nice. And I too just want to start by acknowledging the Wurundjeri people of the Kulin Nation. We're really privileged to be able to practice our trade here. So my respects to the elders and any other First Nations people here today. So today I'm going to talk about my work on characterising the chromatin protein Smchd1 and using genomics to try and understand the molecular mechanisms by which it's involved in regulating transcription, and then sort of validation, where I start to get down to more single cell level to validate those techniques and what we found. So the reason I'm interested in Smchd1 is because it is involved in regulating the DNA, and the regulation of DNA throughout development is really important. So what I'm showing up here is DAPI staining the DNA in a pre-implantation embryo. And what I hope you can appreciate is that the structure and the distribution of the DNA changes really quite dramatically, even in just these early stages of development. And this continues well beyond pre-implantation development in the embryo in such a way that a single genome or cells that possess the same copy of the single genome can give rise to broadly different functions with really different and distinct transcriptional profiles. So we're interested in the molecular mechanisms by which DNA is regulated. And we know that DNA doesn't actually exist as a naked molecule within the cell. It's packaged into proteins, and DNA and these proteins associated are referred to as chromatin. So I'm showing here, this is the nucleosome, which is made up of a histone octamer and the DNA that's associated with it, and I'll be talking about this quite a lot. So the chromatin has the capacity to be covalently modified. So the DNA itself can be modified. I'm showing here, for example, DNA methylation of CpG dinucleotides, but also the N-terminal tails of the histone proteins can be modified. And these modifications serve to regulate the contacts made between the DNA and the histone proteins or associated proteins. So you can get modifications, such as the ones shown in green, that are associated with more loosely packaged chromatin or euchromatin, and these are generally associated with active transcription. And you get modifications shown here in red that are associated with more tightly packaged chromatin or heterochromatin. And these areas are generally transcriptionally or more transcriptionally inert. And I've also depicted DNA methylation here, which is associated with transcriptional repression, as black fields in circles and white fields in circles will generally mean DNA hypomethylation. So these covalent modifications are catalyzed by and then bound and read and interpreted by a host of proteins that are referred to as epigenetic modifiers. And these act to maintain or propagate a particular chromatin and thus transcriptional state. And I think this maintenance is a really important point because by definition, epigenetic silencing and epigenetic regulation is mitotically heritable, so maintained throughout cell divisions. So what we've gained more of an appreciation of in recent years is it's not just the local chromatin architecture that's important for regulating transcription, but also a higher order chromatin structure. So interactions between, long-range interactions between bits of DNA that aren't actually close together in linear sequence. So most people should be familiar with the concept of DNA looping. So promoters interact with distal DNA enhancer elements, and they come together in the nucleus to regulate transcription, normally to promote transcription. And this is mediated by proteins that create these looping events. You also find these domains, which are made up of lots of looping events that tend to occur more frequently with each other, creating this topologically associated domain. And one, or I'll call them TADs throughout the talk. So one TAD tends to interact with itself more frequently than another TAD, and there's very little overlap or interaction between the two. If you look at the next sort of level up in the sort of hierarchy of genome organization, you can define these subcompartments. So A and B compartments are probably the most well-known compartments, or one of the earliest defined ones. And these actually associate or correlate with transcription. A compartments are generally the more euchromatic, transcriptionally active regions, and B compartments are the heterochromatic and more transcriptionally inert regions. So nuclear positioning, so right up at the level of the nucleus, is also really important for regulating transcription. So what I'm showing here is a nucleus with all the different chromosomes stained with chromosome paints. And you can hopefully see that each of the chromosomes occupies a kind of discrete territory with very little overlap within the nucleus. But we also know positioning relative to other nuclear features is really important for regulating transcription as well. So genes or regions that are close to the nuclear periphery tend to be silenced and more heterochromatic. So none of the modifications that I've just described really work on their own. They work in concert to regulate transcription or a particular chromatin state. And a really dramatic example of all of these features being used together is X chromosome inactivation. So this is a process that occurs in female mammalian cells that have two X chromosomes, and it's evolved as a dosage compensation mechanism. So in pluripotent cells, which you can think of as kind of epigenetically naive, they have two active X chromosomes that are transcriptionally active. And as the cells begin to differentiate or development ensues, the initiation of silencing is sort of marked or started by this upregulation of EXIST, which is a long non-coding RNA, which actually binds and coats the future inactive X chromosome in cis and initiates this silencing cascade. Silencing is then established by the exclusion of RNA polymerase II and accumulation of repressive histone modifications at the same time while losing active histone modifications. And as well, the inactive X chromosome is recruited to the nuclear periphery. Silencing is maintained, and this is important, maintained through subsequent cell divisions throughout the lifetime of the female organism. This is marked by the recruitment of Smchd1, so the protein that I work on, as well as the accumulation of DNA methylation. So X chromosome inactivation is a very important developmental process, but it is also a really wonderful model system to study epigenetic silencing, so identifying new mechanisms or characterizing proteins that are involved in transcriptional silencing that applies genome-wide. So I mentioned Smchd1, which is involved in the maintenance of silencing. This is the protein that I've been working on for my PhD. So it's involved in X chromosome activation. It directly binds the inactive X. And homozygous null mutations for Smchd1 result in a female-specific embryonic lethality mid-gestation. So suggesting it is really what's critical for the maintenance of silencing on the inactive X. Smchd1 is also known to be involved in repeat-induced silencing in both mouse and human. It's critical in these processes, but also at clustered and repetitive genes throughout the genome. Smchd1 also binds and regulates a number of autosomal loci as well, and I guess mainly imprinted loci as well that are expressed in a monoallelic fashion. So Smchd1 is developmentally important, but also more recently it's been implicated in human disease. So we've known actually for a little while now that loss of function mutations in Smchd1 result in the pathogenesis of a muscular dystrophy, facioscapulohumeral muscular dystrophy, and this is the third most common muscular dystrophy. And so I'm showing a schematic of the Smchd1 protein here. It has a structural maintenance of chromosomes hinge domain and an active, catalytically active ATPase domain. And the mutations that arise in FSHD are shown up the top in red. And what I hope you can appreciate is that they occur along the length of the protein. And these are loss of function mutations. But very recently, our lab's actually been involved in characterizing mutations that are restricted to just this ATPase domain. And these are found in patients that have this congenital birth defect where they're born with no nose and really small eyes, known as Bosma-Irania microphthalmia syndrome, or BAMS. And these appear to arise due to a gain or enhanced Smchd1 function. So there's been a lot more interest in trying to understand how Smchd1 works. And I think particularly a lot of work in characterizing the mechanisms, the molecular mechanisms for Smchd1 function have informed our studies and understanding of FSHD. So this is what the Blewett Lab, many of us in the Blewett Lab are interested in, is understanding the molecular mechanisms by which Smchd1 regulates transcription. And as Marnie said, my particular focus has been on the chromatin, so trying to understand or better understand how Smchd1 interacts with and alters the chromatin. So when I was started, what was known is that Smchd1 actually directly binds to the chromatin at target loci. And from loss of function studies, in the absence of Smchd1, you get an upregulation of target genes. And this is associated with a DNA hypomethylation, so a loss of the CpG methylation, and an increase in the modification that's associated with active promoters H3K4 di and trimethylation. But this definitely wasn't enough to explain, this understanding wasn't enough to explain what we see and the effects that we see in the absence of Smchd1. And we don't really know whether they're a direct or indirect cause or consequence of loss of Smchd1. So with that, the aims of my PhD were to understand, or determine whether Smchd1 directly interacts with RNA on the inactive X chromosome, and that will hopefully become clear in a minute as to why. And what I'll talk about later is investigating the role of Smchd1 in mediating higher order chromatin organization. So when I started my honours with Marnie, my question was to try and determine how Smchd1 was recruited to target loci. And we had people, or Kaylin in our lab, who was taking a more traditional biochemical approach to look for direct interactors of Smchd1. So I was going to take a more functional approach to target this question. So at the time, it was not known how Smchd1 was recruited to any target loci. Since then, it's been found that Smchd1 is recruited to repetitive DNA through this LRIF1 HP1 gamma K9 trimethylation pathway. But myself and others have shown that this isn't important for Smchd1 recruitment to the inactive X chromosome, and it definitely can't explain Smchd1 recruitment to autosomal regulatory regions. So for my honours project and also for the rest of this aim in my PhD, I really relied heavily on the use of cells that possess this Smchd1 GFP knock-in allele. So it produces a functionally wild-type Smchd1 GFP fusion protein with a C-terminal GFP tag. Females are viable, importantly, and it maintains its chromosomal binding, or its chromatin binding. So you can see here, this is an image of the Smchd1 GFP, or cells that carry a Smchd1 GFP allele. And it's enriched as this bright nuclear focus, which co-localizes with the DAPI-dense region, and also H2AK119 ubiquitination, which is a marker of the inactive X chromosome. So the idea behind my honours screen, which I have to quickly introduce, is basically that I would pick candidate epigenetic modifiers that were involved in X chromosome inactivation and knock them down using hairpins and screen for a loss of Smchd1 GFP focal enrichment from the inactive X chromosome. And so the hit that I got in honours year was when I knocked down H and R and PU, we actually lost Smchd1 GFP focal enrichment in a number of cells. And I spent the rest of that year following up a direct interaction between Smchd1 and H and R and PU, and found that no evidence for a direct interaction. But what we actually know about H and R and PU is that it binds to the inactive X chromosome through one domain, and also binds to this long non-coding RNA exist, which I said was important for X chromosome inactivation. And it actually tethers it along the X chromosome, or the inactive X chromosome. So we found that in the absence, so when we knocked down H and R and PU as well in our cells, we did lose exist. So then the hypothesis came that Smchd1 had an exist-dependent localization, or dependency on exist for its inactive X localization. So exist is really tricky to knock down. So what we did is, I took locked nucleic acid probes that are antisense for exist, and you can transfect these into cells, and they directly displace exist. So it was quite a high background experiment, but what you can hopefully see, so the nucleus is circled, and anything outside of that is lysosomal uptake of the probe. But upon transfection with a scrambled probe, the Smchd1 GFP foci were maintained, and when we were targeting exist directly, we lost Smchd1 GFP localization. So showing that Smchd1 has a dependence both indirectly and directly, showing these both indirectly and directly, a dependence on exist for its localization to the inactive X chromosome. So around about the same time, Kaylin in our lab was doing some work with recombinant Smchd1 protein and showed that in vitro, the hinge domain has the capacity to bind to RNA oligonucleotides, and quite importantly, that this function was abrogated when a mutation that's found in FSHD patients was introduced into the recombinant protein. And also, when I was doing these experiments, what came out was a big screen looking for direct protein interactors of exist, and Smchd1 was actually one of the candidates in that screen. So we hypothesized that Smchd1 then might function as an RNA binding protein and directly interact with RNA. So to look at this on a genome-wide level, so to not just investigate exist, but see if this is a mechanism that could exist elsewhere, I performed PARCLIP. So this is a technique that's similar to CHIP. So it involves UV cross-linking of nucleic acids to protein, an IP of your protein of interest, really stringent purification steps because RNA is messy, and then you make libraries and sequence for binding partners. So I performed a GFP immunoprecipitation in female Smchd1 GFP neural stem cells, and I used a no cross-link control to control for non-specific associations with proteins and RNAs in the lysate, and also a wild-type control to act as an isotype control. So this is a Western following the GFP IP, and this was my test and the two negative controls. So we're getting nice enrichment of Smchd1 following GFP immunoprecipitation in the Smchd1 GFP cells, but not in the wild-type controls. And this is an autoradiograph showing the associated RNA that came down with Smchd1 or the two controls. And there was a discernible smear of RNA that came down with Smchd1, and the signal was a lot stronger in the GFP IP than the two negative controls. So we went ahead and made libraries and sequenced these. I mapped the reads, and they were distributed throughout the genome. And then I used a program called Paralyzer to call clusters, which I guess is like chip peaks and identifying binding partners or potential binding partners, and got 4,763. However, when I normalized the background and subtracted the background from either the no cross-link or wild-type control, none of those clusters or none of those reads were maintained. So this suggests that Smchd1 does actually not bind to endogenous RNA in female cells. Obviously, wanted to validate this. So the validations that I'm showing today are focusing on exist because it was our strongest candidate, and also there was some belief in the literature that Smchd1 and exist directly interact. So I did an RNA immunoprecipitation for GFP again, and then could look at the associated RNAs, candidate RNAs by qPCR. And I also performed the reciprocal experiment, IDRIP, where you can enrich for exist using an antisense oligonucleotide, or biotinylated one, and then perform a Western blot for candidate proteins. So these experiments were performed with UV cross-linking, and I also used HNRNPU as a positive control because we know it does bind to exist. So with the RIP, what you can hopefully see is that following GFP, immunoprecipitation of Smchd1, there was no enrichment of exist, nor especially above, or not really, even with the housekeeping controls. But there was a nice and specific enrichment of exist upon HNRNPU pull down. And the results with the IDRIP were consistent. When we enriched for exist, we were not able to detect Smchd1, but we could for HNRNPU, so in support of the two protein Smchd1 and exist not interacting directly. What I was able to do more recently was to get some data in individual nuclei by performing immunofluorescence for Smchd1 GFP, an exist RNA fish, and performing confocal microscopy using the ARIEScan detection. So this is not a single molecule resolution technology, but it does allow you to see much higher resolution, the binding of, or the localization of these proteins within the nucleus, and specifically what we were interested in was the region over the inactive X territory where exist is localized. And what we saw was that, so the images I showed you before, the wide field images, Smchd1 GFP appears to be enriched over the entire X chromosome, but when you can increase the resolution, you actually see that it's localized to these specific domains. And these domains show very, very little overlap, so 7.6% with exist at all, in support of them not actually interacting directly within the nucleus or on the inactive X chromosome. So our data supported that there was no direct interaction between Smchd1 and exist, but it remains still that Smchd1 localization to the inactive X chromosome was still exist dependent. So in the time that I was doing all these experiments, 3 big screens came out that were actually trying to identify novel direct interactors of exist, protein parters of exist, and that included the one that identified Smchd1 as a candidate. So what we did was we took the common hits from these 3 screens and then revisited my original localization screen, so knocking down Smchd1 and looking for effects to its localization. And what I found is actually that knockdown of another HNRNP protein, HNRNPK, actually resulted in loss of Smchd1 GFP focal enrichment from the inactive X. So obviously wanted to investigate whether this was a direct interaction first. And so by performing immunofluorescence for the 2 proteins, you can see they actually have quite a distinct nuclear profile for their localization, but they are both over the territory of the inactive X chromosome. But by performing immunoprecipitations and looking to see whether the 2 proteins co-IP in the whole cell extract, but also the pellet that's been treated with nuclease and as well as high salt treatment to release the inactive X or the heterochromatin from the insoluble material, IP of Smchd1 did not result in, you didn't co-immunoprecipitate with HNRNPK in either fraction, and we did not get Smchd1 co-immunoprecipitating with HNRNPK as well in either fraction, which is in support of them not directly interacting. But this actually fits with all of the proteomics data we've had over the years where we haven't found direct interactions with Smchd1 at all. And I think it's led me to this model. So what I've shown you today is that Smchd1's localization on the inactive X chromosome is dependent on the long non-coding RNA exist and also its downstream interactor HNRNPK, but they don't actually directly interact. But what HNRNPK has been shown to be important for on the inactive X chromosome is the recruitment of these 2 complexes, polycomb complexes, PRC2 and PRC1. So in my honours year, I actually did a bit of work investigating a potential interaction and dependency on PRC2 for Smchd1 localization by both knockdown and deletion, and wasn't able to find any dependency, but I haven't really investigated PRC1 at all. And what we know about PRC1 is that it actually modifies the chromatin. So it catalyzes this mark, H2A lysine 119 ubiquitination, but it also has a role in mediating these higher order chromatin structures and these repressive bodies known as polycomb bodies. So what I hypothesize is that Smchd1, rather than being targeted to the chromatin by a specific protein-protein interaction or RNA interaction, that it could actually be recognizing a specific feature of the chromatin. And this is something we wouldn't have been able to detect by any of the assays that we've been doing, the large-scale proteomics or genomics assays. So to follow this up, we're going to do some ChIP-seq for Smchd1 on the inactive X with Andrew Kennery, and also some immunofluorescence studies for various chromatin modifications and see if we can validate this model. So this leads me to my next aim, which is to investigate the role of Smchd1 itself in mediating higher order chromatin organization. There's some hints that Smchd1 might be involved in these processes from its structure alone and also from a few little bits of data in the literature. So this is the same schematic before. Smchd1 has C-terminal SMC, or structural maintenance chromosomes domain, which is homologous or shares homology with proteins that are really critical in driving these looping and TAD processes. And so there's been some data in humans to suggest that Smchd1 on this end of this sort of scale has a role in the compaction of the human inactive X chromosome. And this has been shown by immunofluorescence and imaging, where in the absence of Smchd1, you get a decompaction of the inactive X in humans. But data from our lab has actually kind of raised the possibility of Smchd1 being involved in processes more down this end of the spectrum. So there's this possible functional opposition that exists between CTCF, a protein that's particularly important for these events, and this is based on sequencing, on expression data and also on Smchd1 occupancy. So for the experiments that I'll describe in this part of the aim, I actually wanted to do these experiments in female cells because I was interested in the inactive X chromosome. But remember before I said that if you have no Smchd1, it results in a female-specific embryonic lethality. So to get around this, we used a conditional allele to delete Smchd1. So we derived cells that had a conditional allele that had LoxB sites around exon 4. And then once the cells were in culture, could use Cre-mediated recombination to delete Smchd1. And 1 week, or 5 days and 1 week post-Cre-mediated recombination, there was no Smchd1 detectable at the protein level. So we subject these cells to RNA-seq and found that there were 1,683 differentially expressed genes between the Smchd1 deleted and flock cells. So this first showed that you can actually lose Smchd1 in a cell that's already gone through the maintenance of and also the establishment of silencing and still get a change of gene expression, implicated it in the maintenance of gene expression as well. So this is really the first time we've looked at deleting Smchd1 in cells rather than just looking at cells that have never had Smchd1. So Matt Ritchie had previously done some gene expression analysis on cells that are derived from mice that have a null allele of Smchd1, so have never had Smchd1. So he was able to use my top table to do a gene set enrichment test and find that there was concordance between the Smchd1 deleted and Smchd1 mummy D1, or the null allele of Smchd1 cells possessing those. So together, this indicated that these cells would be a good model to study Smchd1 function. So to look at Smchd1's role in mediating higher order chromatin organization, I performed in situ HoxB. So this is a technique where you cross-link your DNA and your proteins within the nucleus and try and lock everything in its position. And then you perform a restriction digest and you cut the DNA. You then fill the ends with biotin and perform a religation. And what hopefully you can see from this schematic is that these two bits of DNA that were not actually next to each other in linear sequence at all, but were being held together by a protein interaction or a protein interaction with the DNA, have now been brought together, and you produce this chimeric bit of DNA. You can then isolate those bits of DNA and subject them to deep sequencing, and this allows you to map and find those bits of DNA that are interacting in physical proximity, but not necessarily in linear sequence. So I mapped and filtered these reads using Hiccup, which is a pipeline designed to treat these tricky chimeric reads, and then performed a differential analysis using diff-IC. And this is actually developed here at WEHI, and it uses edge-R statistics to perform a differential analysis. So in this case, between the females with the flox allele and the deleted allele, or that had Smchd1 deleted. So one of the parameters that is really important to choose for this sort of analysis is your window size. So you bin your reads. And so most people they can do down to sort of 40 kb reliably with the sort of coverage that we got. But I'm presenting data today from my first pass analysis, which is looking at 1 megabase windows, but I've also done the analysis at 100 kb and seen consistent results. So from this analysis, I found 2,384 differentially interacting regions between the Smchd1 floxed and deleted cells. And it's quite comforting to see that most of these actually fell or were anchored on a region where we can detect as direct Smchd1 binding set by ChIP-seq. So it was 85% of those. And these were over regions that we know that Smchd1 is either heavily bound or has a strong role in regulating transcription. So at the protocadherins, in printed clusters, the HoX clusters, and of course the X chromosome. So I'm going to tell you about sort of one of the top hit, actually, in that top table, and that came up a number of times. But before, I will have to explain how Hi-C data is shown. So this Hi-C data is depicted as a heat map. And on each axis, you have a region of the genome. The points on there are a reflective or a representation of the interactions that you detect via sequencing, and the strength or how dark they are indicates the strength of that interaction or how often it was detected. So along this diagonal axis, or along the diagonal, this sort of represents your linear genome. And you can see that the signal is strongest along your linear genome, and this has been shown by many people, and this is what you'd expect. But if you move away from the diagonal, you can see these points that are still dark and being detected as regions of where there are interactions. And this region here represents an interaction between this bit of DNA and this bit of DNA that they're capturing by this technique. So this here is a heat map from the wild type cells, or the flox cells. And this is raw data. But what I've shown in the boxes are the regions that are getting called as that were in the top 10. These regions are in the top 10 differential interactions between the Smchd1 floxed and deleted cells. So when I looked at this region in more detail, what we could see is that they were anchored around the HoxB cluster. And this is some ChIP-seq, or a track from ChIP-seq, showing that Smchd1 actually really strongly binds to the HoxB cluster. So 22 of those differential interactions were being lost from the HoxB cluster upon Smchd1 deletion. So if we have a look at the top interaction, differential interaction, you can see the box here is around the region that was called as differentially interacting. In the Smchd1 floxed and the Smchd1 deleted heat maps shown down here. So one end of this interaction is anchored in the HoxB cluster, and the other end is anchored in this keratin cluster, so another region of repetitive genes that's directly bound by Smchd1. And if you have a look here, you can see the interactions that we're losing upon Smchd1 deletion. So round about the same time that I was doing this, Tam in our lab was actually preparing, doing some stains of embryo skeletons or embryonic skeletons that were either wild type or homozygous or had the null allele of Smchd1. And she was actually, I think she'd been looking at lots of things, but she observed that in these embryos that had no Smchd1, there were phenotypes that were consistent with a dysregulation of HoX genes. So this embryo is missing a rib, and she's observed that a few times, and there's also been a polydactyly observed that are classic homeotic phenotypes. So for those of you that aren't familiar with HoX genes, they are really important in the patterning of bilateral organisms. And the HoX genes in mouse exist in 4 paralogous clusters. There's 56 genes between them. And the order in which they appear in the genome reflects the order in which they're expressed in the developing embryo in both a spatial and temporal way. We have never seen a disruption of HoX gene regulation in any of our RNA-seq data sets, but I think it's because we've been looking in the wrong tissues. So we've mostly done whole embryos or cells made from this part of the brain that don't express HoX genes. So what we're going to see what we're going to do to see if Smchd1 has a role in regulating Smchd1 and try and understand, in regulating the HoX genes, and also see if we can try and understand what these loss of the chromatin structure means, is to actually perform an in vitro differentiation of embryonic stem cells, pushing them down into neuromesodermal progenitor cells. And this is just a schematic here depicting what happens, but you basically get activation and repression of HoX genes along the cluster over the differentiation time. And so this is the most sensitive way to look at HoX gene expression, using an in vitro way, and it will kind of inform our studies with embryos later. And we're actually starting this tomorrow. So back to the high-C data. So I mentioned I was really interested in the X chromosome, and obviously Smchd1 has a prominent role in X chromosome inactivation. And actually a lot, so 516 out of the 2,300 odd differential interactions did map to the inactive X chromosome, or the X chromosome rather. So I'm showing the regions that were called as differentially interacting mapped along the X chromosome. And I'm just showing you chromosome 2 as an autosome for comparison. You can see they really are more heavily enriched and really distributed along the length of the chromosome. So the first thing that I really wanted to look at, given that Smchd1's been given a role in the compaction of the human X chromosome, is whether we see the same sort of thing in mouse. So a loss of compaction upon Smchd1 deletion. So to do this, I used a chromosome paint to paint the X chromosome in nuclei that either were wild type or deleted for Smchd1. And then I was able to use, again, the confocal microscopy with Eriscan and measure the volume in 3D. So there was no difference between so we're not able to discern between the active and the inactive X, but what has been established in the field over a long time is that the inactive X is on average 30% smaller than the active X, and this is what we saw in each nuclei as well, one that was about 30% smaller than the active on average, than the biggest one on average. And if I looked at the difference in each nuclei between the inactive, or the biggest, and the smallest X volume, either as a difference or percentage, there was no difference upon Smchd1 deletion, suggesting that in mouse, you do not see a loss of compaction of the X chromosome upon Smchd1 deletion. But what I also found, and this was kind of by accident, because I was staining for this modification of the chromatin, H3K27 trimethylation, and this is a marker of the inactive X chromosome. And I was kind of doing this to get around doing this experiment as a kind of cheap and easy way to do it. But what I observed, and you could even see just on wide field microscopy or images from wide field microscopy, is that this focus on the inactive X actually becomes much, much stronger upon Smchd1 deletion. And we could see, by quantitating this, both an increase in the volume of this K27 trimethylation staining and also in the intensity. So this modification is another repressive modification, or a modification that's associated with gene silencing, and could, in one part, explain why we're not seeing any massive changes in compaction upon Smchd1 deletion. But Andrew Kennery has performed some chip for this mark as well, and has seen the same thing, but has also seen a spreading of this modification into regions where it's not normally found. So this kind of raises the possibility that maybe Smchd1's having some sort of insulating role in protecting different chromatin states from spreading where they should normally be. So another way that you can look at compaction and accessibility of a region of the genome is to perform a TAC-seq. So basically the reads that you get from a TAC-seq, you can quantify, and it's a way of really looking at how open or closed a particular region is. So the more reads you get in a region, the more open that chromatin is. So I performed a TAC-seq, called peaks, and did a differential analysis of those peaks. And looking on the X chromosome, I've shown all the peaks that have been called as differential between the Smchd1 deleted and the Smchd1 fluxed. And sort of consistent with the data that I got from the imaging, there's not massive changes to the compaction overall of the length of the X chromosome, but the differential regions seem to be coming up in these clusters. And these overlap with what I found to be later, these large tandem repeat regions that have been defined on the X chromosome. So upon Smchd1 deletion, you get increased accessibility at these large tandem repeat regions on the X. So these large tandem repeats, or one of these large tandem repeats actually, DXZ4, has actually been found to have a really important role in maintaining the structure of the inactive X chromosome. So this is someone else's HiC data, but what you can hopefully see, so this is the active X chromosome, and this is the inactive X chromosome. And you can see there's a lot more interactions that come off from the diagonal, so a lot more structure on the active X chromosome compared to the inactive X chromosome. And the structure that is on the X chromosome, really on the inactive X chromosome, is based or centered around this one point here, which is the DXZ4 tandem repeat array. So I think putting all of that data together has, I know that I didn't say this before, but when we delete Smchd1 on the active X chromosome, you get increased interactions from HiC. So what I'm thinking is that maybe Smchd1 has a role in binding and regulating these tandem repeat regions that are important for structure on the inactive X chromosome. And when you lose Smchd1, you're getting enhanced interactions, so maybe the chromosome is adopting a more active-like structure because it normally plays a role in insulating these interactions. So in a couple of slides, I'll talk about how we're going to continue to look at this and validate this model. But I also wanted to see if the attack-seq data that we generated could tell us anything about what Smchd1 might be doing mechanistically to the chromatin on the autosomes as well. So I said before I did a differential analysis of the MAX2 called peaks from the attack-seq data and found 4,205 differentially accessible regions. And then filtered the list for annotation for those where we saw the greatest effect of loss of Smchd1. So this is showing the values for all the peaks, so the coverage for all the peaks, and in the Smchd1 deleted and the floxed. And there was no massive overall change to the accessibility upon Smchd1 deletion. But if you just look at these differential peaks, so these are the Smchd1 deleted and these are flox ones, you can see the values go up, indicating that these regions you have an increase in accessibility upon Smchd1 deletion. So if I looked at where these peaks sat relative to the transcriptional start site, normally your attack-seq peaks, or the vast majority of your attack-seq peaks, sit at the promoter and at the TSS, whereas these differential regions actually sat quite distal to the transcriptional start site, up to sort of the majority of them 50 to 500 kb away. So I was annotating these peaks and I looked at a lot of features, but what I found is that these differential peaks are actually enriched for neural enhancers. And this is consistent with what we found from chip sequencing data. So Kaylin's found that Smchd1 actually binds at enhancer regions. And this is where we're seeing changes to the chromatin accessibility. But this was only in 132 of our 913 differential peaks. But it's working. If anyone's worked with enhancers, they're quite tricky to get good annotations for them because they're very tissue-specific and developmental stage-specific, and there's lots of them, and they're very far away from the genes. So to look at this in a more unbiased way, this is showing some chip sequencing data that Andrew Kennery performed. So what he did was chip sequencing for H3K27 acetylation, which is a mark of active enhancers, and as well H3K27 trimethylation, which is found at enhancers that are inactive or poised. So these are the Smchd1 flox cells. This is showing the reads over those differential attack peaks for the chip-seq reads over those regions that were different in the attack-seq. And what I hope you can see is that you've got, upon Smchd1 deletion, an increase in this K27 acetyl mark, which is a marker of active enhancers. And maybe a slight loss of H3K27 trimethylation at these same regions, which suggests Smchd1 deletion results in the ectopic activation of distal enhancers. So I think taking all these data together, it's led me to this model that I want to spend my rest of the time, or the rest of my time in the Blewett lab sort of testing. But I propose that Smchd1 actually mediates long-range interactions between repetitive and clustered regions of DNA, creating a silent domain or contributing to a silent domain, which limits promoter-enhancer contacts, so by insulating these events that would occur without Smchd1. So in the absence of Smchd1, you get these events, these promoter-enhancer interactions, which are permissive for transcription. So what I want to do to kind of follow up that model and try and validate it while I'm still here is to actually further analyze the HiC data that we've got. So the analysis that I've done with diff-hic has been really wonderful and really powerful for looking at the differences, but it doesn't allow you to model the actual chromatin, the changes to the chromatin in the absence of Smchd1. So not really allowing us to place what we're seeing within this hierarchy of chromatin organization. And particularly for the inactive X, where we're interested in that really well-defined structure. I also can probably ask with the data that we've already got, are there differential chromatin interactions at enhancers that are activated upon Smchd1 deletion, and correlate this with transcriptional data? I want to do some independent validation of the HiC, and also looking at what you're seeing in individual cells and individual nuclei, by performing DNA FISH, followed by super resolution imaging at the HoxB cluster, and also the inactive X chromosome. And this will allow us both to discern the inactive X chromosome and look at things more, look at conformation and organization beyond just the volume analysis that I've done so far. And then I want to do a few more HiC experiments, investigating whether Smchd1 might have a direct role that will hopefully let us get whether Smchd1 might have a direct role in mediating these changes that we're seeing. So kind of a rescue experiment. So does the reintroduction of Smchd1 restore the chromatin architecture? And this has been done with other proteins before, so there is sort of proof of principle that this can be done. And look at various different mutants. So we've got mutations in Smchd1 that enhance the ATPase activity and ones, or enhance the silencing function of Smchd1. And we're making a catalytically dead ATPase domain in Smchd1. So seeing what the role of the ATPase domain is in mediating higher order chromatin structure. So with that just leaves me with my acknowledgements. So Marnie Blewett and James Murphy are my supervisors. They've been the most wonderful supervisors. I know everyone says that, but they are. And they have given me absolute free rein with my project to do anything I want, really. I've done a ridiculous amount of tricky techniques, so thank you for having so much faith in me and letting me do that. Warren, Joan, and Andrew are my PhD advisory committee, and they've been nothing but really kind and supportive for the last 3.5 years. Everyone in the Blewett lab, so I put Andrew Kennery again because he's helped with the chip that I've shown today, but he's also been a wonderful mentor and friend, so thanks for the great chats. Kaylin Chen, she's just left our lab, but she worked on Smchd1 before me, so she did the PhD on Smchd1 really before me, and set the groundwork for my project, and everyone in the lab really changed the direction, which was awesome. Kelsey has derived all the neural stem cells I talked about today that I used in all my experiments, and she also helped me culture the billions and billions of cells that I needed for all the genomics, so thanks. Tam did the skeletal preps that I showed you earlier on, where we saw the homeotic transformations or potential homeotic transformations, and both Meg and Kelsey helped me set up the screen where I found HNR and PK as a potential interactor of Smchd1. Everyone else in the Blewett lab has been really, really supportive and helpful over the years, both with ideas and just in the lab whenever I need it. The molecular medicine division is a really wonderful division to work in. It's really fun. Tracy Wilson, special mention, she's done all the cloning for our lab, almost exclusively on the whole division. Andrew and Adrian have done some help with CRISPR before that I didn't show in the talk. And Matt, Charity, and Stuart have always been around when I've been trying to teach myself bioinformatics for any stupid questions I might have. Peter, Hickey, and Hannah have actually both helped me a lot and been there to advise me on all the genomics analysis that I've done as well. And Hannah's the sort of resident HiC expert and has been really helpful. Stephen's facilitated all the genomics that I've performed. Kelly, Niall, and Lachlan have helped with all the imaging and the analysis. Tim and Reese provided me with the in situ HiC protocol. Of course, bioservices for all their help with all the mice along the way. Graham Kay produced the Smchd1 GFP knock-in allele that I used. Archer and Simon took me over to the Perkins Institute in WA and taught me parclip. Edwina McGlynn we're going to be collaborating with to look at Smchd1 in its role in regulating HoX genes. And of course my family and friends. So I've got my whole family here today, which is really, really nice, from New South Wales. And all my friends have been really, really supportive and kept me sane over the last 3.5 years. So thank you. Thanks, Manny. Thank you. Cool.
Okay, questions? David?
Do you think Smchd1 affects HoxB expression or function?
So the expression of the genes or the function of the protein?
HoX itself, or is it affecting genes that are affected by HoxB?
Well, I think they kind of go hand in hand. So if you affect HoxB expression, then they're not going to be able to affect their downstream targets, or we'll have more. So what I hypothesize is that, and we'll hopefully see with this differentiation, is that when you lose Smchd1, you lose the maintenance of silencing of HoX genes, so it's really important to turn them off in subsequent stages of development. So maybe you're getting ectopic activation of the HoX genes, and then they can go and act on the downstream targets. So they kind of go hand in hand.
One of the very first really nice large nuclei where you showed us Smchd1 GFP co-localizing with the inactive X. There was another Smchd1 GFP focus in that.
Yeah, so I didn't say that, but a lot of the experiments, so the imaging experiments and the early ones I did in my honours were done in immortalised MEFs, and they're tetraploid, so have 2 inactive X chromosomes. Yeah, Tim.
Some of the interactions you showed in your examples are very long range.
Yeah.
I wonder if you could say, tell us the average span of your DIs or the breadth between the 2 anchors, and what do you think might be the nature of those sort of very, very long interactions?
Yeah, absolutely. So those, I don't off the top of the head know the average range for those ones at the 1 megabase resolution, but what I think is that Smchd1 generally binds to regions that it's really important for them to be switched off throughout development, so they're developmentally important regions. So potentially, and a lot of them are really clustered and repetitive, so I showed the keratin and the HoX genes, but it's also bound at like the olfactory receptors, and cluster protocoterins, for example. So potentially it's involved in creating sort of a repressive domain similar to the sort of polycomb bodies, or potentially they are related to the polycomb bodies, so something like that where it's really, really stable, dense silencing, and it might not happen with a huge frequency, so you can see the interactions, they're not as strong as some that you see by high C, but yeah, I think it's potentially a really important stable silencing mechanism to really switch these genes off.
You didn't appear to have time to discuss your proposed function of Smchd1 in the context of the clinical outcomes of the loss of function mutation.
Yeah, absolutely. So I think in FSHD, at least we know that Smchd1, so I talked about the DXZ4 locus on the X chromosome. In humans, Smchd1 binds to a similar autosomal repeat region, D4Z4, is important for silencing that locus. So whether this is through mediating higher order chromatin changes, and this might be an energy-dependent process. So what other people in the lab are doing, and Marnie's working on, is doing sort of drug screens to modulate the ATPase activity of Smchd1. So potentially if, so the future high C experiments we do and other biochemical experiments show that the ATPase domain is important in Smchd1's repressive function, potentially its role in silencing these repetitive clusters can be enhanced with a drug treatment if we understand that that's how it's working, hopefully.
So in the experiment where you inhibited exist function, and I forgot what you did, but you
Yeah, the locked nucleic acids.
How would you distinguish, I mean, do you know what that does to actually exosomal activation? Maybe you don't have an inactiveness in your cells anymore.
Yeah, so you can actually, so silencing of the inactive X chromosome is incredibly stable. Once it's in the maintenance phase, it's really hard to get reactivation. And it's been shown that loss of exist doesn't, it's important later on, but it is kind of dispensable in the maintenance phase of X inactivation. So you do have gene silencing. And what I didn't show and what other people have shown is that when you transfect the cells with these locked nucleic acids that displace exist, you're still getting exist expression. It's physically displacing the copies that are there. So over time, you actually get reaccumulation of exist that coats the inactive X in cis, but I would assume that many of the other chromatin marks are still retained.
Any last burning questions? Okay,
Cool.